this is also organized into two parts, part 1 and part 2. Here in this class, we will cover part 1 only. And what is the objective of our course? It is written here actually in the document of our university that course outcome 1 is just we will describe the measurement procedure of various physiological parameters in connection with biomedical instrumentation. And objective number 2 is we will explain various bioelectric potentials. We'll explain obviously what we mean by all such things. Okay, and we'll see the operating principles of biopotential amplifiers. We'll see that thing also. And we'll illustrate the methodologies for acquisition and processing of biomedical signals. And we'll investigate the suitability of bioinstrumentation system to various physiological events. That is a much later topic, obviously. So that is the course objective. Means, in various universities also, or research organization in our country and especially in the outside of our country, they are asking for one paper to be covered by the student where some elements of biology will be discussed. It is very amazing thing, and we could not imagine this type of thing in our student life also when we were the students. But that is happening nowadays because biology is now having the maximum thrust throughout the world, and the coming decades are actually the decades of biology. It was told that all the old millenniums were the millenniums of physics. Now it is believed that except the area of astrophysics and quantum physics, mainly all thrust will be shifted from physics to the biologies. Okay, personally, I did not like biology in my student life at all. But my orientation and focus interest have been changed a lot. Now I like this subject very much, although I don't know too much on biology. Only some aspects that are related to our engineering. Okay, I try to study these things till now. So let's start our discussion on our subject, special paper 3. Just 1 minute. Special paper 3, that is biomedical instrumentation. Can you see all such things? Am I audible?
Yes, sir.
Okay, let's start. And one more thing I should say before starting our class, that whatever thing we will discuss here in the presentation, I will give a PDF copy also to you after the class. Okay, so that will be circulated. So just you should, if you attend the class, you can just have an overview of the subject. You can have the basic understanding. After that, you can study on your own. You'll get all the PDF copy of this presentation. Now, this is about chapter 1. You see the biomedical instrumentation. Let's see what we actually want to do. These are organized into various chapters. One is the introduction. We'll see apart where some basic aspects will be discussed. Next one will be, we will see the origin of biopotentials. Means in our body actually various voltages are generated within the cells and outside the cell, in between the cells also. So how the voltages are generated, the basic principles we'll see. In the chapter 3, we'll see that all these voltages will be added up, will be summed up, and ultimately there will be a manifestation of cell level voltage into the surface. Okay, various surface of our organ. Okay, a surface of the skin, various parts of the body. These are called the surface potential. So from cell level biopotential, biovoltage will switch to surface level voltage gradually. Then we'll see how to collect these voltages. These voltages from various surfaces of our organ will be collected by a special type of electrodes, not a normal electrode. These are called biopotential electrodes. We'll study this thing here in this class. Then we'll see that there are various sources of noises present in our body. These are called artifacts and noise. There is a difference between artifacts and noise. A noise is a true noise, a noise picked up from the environment, a noise generated within various unknown regions of our body. These are the noise voltages. Okay, we'll see. But artifacts are coming some other signals whose origins are not known or may not be known also in some cases in our body, and that will intervene into the noise, into the signal that we want to study actually. Okay, so artifact is actually the source of other signals. It is the other signals that we don't want to study at all, but these are actually intervening into the situation. Don't worry, I'll discuss this thing later. Then comes the biopotential amplifiers. How these various signals are amplified and how to apply them, how to design them. Then we'll see various biopotential signal conditioning, how to design various circuitry to pick up, to transmit, to process these signals. So this circuitry, some things we'll see this thing. And the chapter 8, that is the acoustic and optical signals as biosignals. In this particular class, we will not study this thing at present. Something will be discussed in the other part of this subject. Here, because there are so many things there up to chapter 7, so we will confine ourselves within such things only. Okay, so let's go to the chapter 1, our first thing. That is the introduction to our biological system. That will be relevant to our particular purpose only. Okay, we don't want to study biology just from the medical perspective. We want to study biology from engineering perspective. That is the difference. Doctors are studying some subject. These are called medical instruments in their medical curriculum, MBBS course. They also study some other things related to the various medical instruments. But these are all from their perspective, medical perspective. And we study biology from engineering perspective. Obviously, in short, as overview only. Let's see this thing now. Now, first we want to see our biopotential, means the voltages that are generated inside the living cells, tissues or organisms. Okay. And why these biovoltages, biopotentials are generated? These voltages are generated due to the ionic transfer, okay, through the cell membrane from outside part of the cell to the inside part of the cell, or in the reverse direction, from inside of the cell to the outside part of the cell. The outside part of the cell is called extracellular medium. The inner part of the cell is called intracellular medium. Okay, so there is an ionic transfer, and the cell membrane will selectively allow them under certain condition so that ion can move, can pass through the membrane, and there will be some change in voltage inside the cell and outside the cell. The cell membrane will have a different voltages at different point of time and different space also, okay, throughout the dimension. So ultimately some electrical signal will be manifested, okay, and these signals will describe some information from which things can be understood, can be diagnosed. All these things will be possible. All features will be found. Okay. And note that why the ions will transport from one point to the another point in the various parts of the body. It is actually as a result of biochemical process. Various chemical reactions, chemical processes will take place inside our body, inside, okay. And as a result of such biochemical processes, there will be some ionic transfer and there will be a change in voltage. So the condition of our cells, tissues or organisms will be reflected in terms of biopotentials, in terms of voltages. So if we study those voltages, will have a lot of ideas, okay, of regarding the condition of our body, condition of our biological system. Mainly the biopotentials occur inside our nerve cell and muscle cell. Okay. And nerve cell it will be found where? Inside the spinal cord. It will be inside our brain. Okay. And it will be throughout the periphery of our body. The nerves are actually branching out to different directions throughout our body. Okay. So it's a all pervasive system. So these biopotentials will occur, will generate inside the nerve cells and will also move, okay, across the nerve cells. And it will be also be found in our muscles. Okay. It will be picked up actually properly. It will be sensed, processed, all such thing will be happening. Now just here is the picture that will show you schematically the condition of our cell. The right side is the intracellular medium. This right side, you see the right side is the intracellular medium, the violet part. It is inside the cell. And the blue part, it is outside of the cell called extracellular medium. And we will find out that some of the ions are mostly present under normal condition inside the cells normally, and some of the ions normally present mostly outside the cell in the extracellular medium. Just like our sodium ions, they are present in the extracellular region outside the cell mostly under normal condition. Our potassium ions, they mostly present inside the cell, okay, mostly present inside the cell, that is in the intracellular medium. The chlorine ion, okay, the negative ion, they mostly present outside the cell, extracellular medium. And okay, some anions, anion means negatively charged other ions. Our chlorine is also an anion. It is one anion. Any negatively charged ion is called an anion, you know. Okay, there are some other anions, they will present inside the cell mainly under normal condition. So because of such separation of charge under normal condition, the cell membrane will attain a voltage. The cell membrane will attain a voltage. Okay. So what voltage all such thing we'll see obviously gradually one by one. Okay. Now you see that since the concentration of ions are different inside the cell and outside the cell for different ions, the ions will have a tendency to move from one part to the other part. Just like you consider that our sodium ion, since its concentration is more in the outside part of the cell, it will try to move into the cell. Similarly, our potassium ions, they will try to, their concentration is more in the intracellular region, so they will try to move towards the extracellular medium. So that is the thing. But whenever they try to move, don't think that it will be possible for them to move. No, it cannot move. The movement, the transfer of ion will be possible only when the cell membrane allows them to transfer at all. Otherwise, movement will not be possible. So it is the role of the cell membrane that will dictate the transport of ion from one point to the another point. Okay. So that is this particular thing. So that the diffusion of ion will take place conditionally through the cell membrane. Okay. Now, how these things will happen, how such movement will be there? There are, okay, the one driving force of the transport of movement will be the concentration gradient. As the sodiums are mostly present, okay, outside the cell, so its concentration is higher in the outside part of the cell, and its concentration is less in the inner part of the cell. So concentration gradient of the sodium ion is higher, concentration is higher in the outside part and lower in the inside part. So there is a concentration gradient means change a slope of concentration curve will be there. That slope will be such that the sodium ion will move from higher gradient to lower gradient, higher concentration to lower concentration. Okay, passing through the cell membrane. Same is true for the potassium ion, but in the reverse direction. The potassium ion, its concentration gradient will be from intracellular to extracellular medium. Okay. Now, mostly the inside of the cell is more negative under normal condition than the outside part of the cell. Okay. It is under normal condition or resting condition. This is called resting membrane potential. Resting membrane potential. And its normal magnitude is negative. Normal value is negative. Get my point? So the resting potential or resting membrane potential is having a negative value. Okay, always for our human body or for most of the living organisms. For most of the living organisms. Okay. Now, if we see in a more closer look, we'll find out that the sodium and chlorine will have higher concentration gradient at the outside part of the cell, and potassium and anions are having some other anions are having higher concentration gradient at the inner part of the cell. Okay. Now, the resting membrane potential, how it is generated? It is because that ions are distributed in a different amount, okay, in the inner and outer part of the cell, and that's why there is a separation of charge, okay, across the cell, and therefore some voltage will be available. And the cell membrane has selective permeability, means it allows some ions to move, to pass through, and it does not allow the other ions to move at a particular point of time. So the permeability of the ion is different. Permeability means the easiness of flow, the easiness of flow of the ions to move from one side to the other side through the membrane. The permeability of ions are different, okay, for different ions, and therefore the ions cannot move equally from one side of the membrane to the other side, and therefore some charge separation will take place and some voltage will be available along the cell membrane. Normally the cell membrane is having negative voltage in the inner side. Okay. So due to the different distribution, actually it is happening, what I told you is written. And selective permeability is happening due to the characteristic feature of the membrane material, and at the same time due to the size of the ions also. Okay. Some of the ions, after getting hydrated, means after getting bonded with the water, is having higher size. So therefore they cannot pass easily. Okay. Just like that, okay, although the atomic weight of potassium is larger than sodium, okay, but the hydrated sodium is having larger size, okay, than the hydrated potassium ion. So the permeability of sodium ion is relatively less, okay, although it is having lower atomic weight, because its size is higher after being bonded with water. Now there is an equation, okay, just I want to show you. Now, if when the class will stop automatically after 10 minutes, don't worry, you already know, still I am saying, click the link, okay, and you'll be back in the class again. So now let's see that there is an equation called Gibbs Donnan membrane equilibrium condition. What is that? First thing is that, first part is that when there are two solutions, okay, in a container, we call it a solution A and a solution B. They are there in that particular thing, okay, and each solution initially is electrically neutral. So number of the cations, cation means positive ion, you know, must be equal to the number of anions in one solution. Then only they will be electrically neutral. So the number of cations in solution A must be equal to the number of anions in the solution A, which is written here. Cations of A is equal to anions of A. Same is true for solution B, where the number of cations is equal to the number of anions in solution B. So that neutrality will be always maintained. Okay, when two solutions are separated by some membrane, some permeable membrane. Now another thing is that some of the ions that can diffuse to the membrane, okay, from one side of the membrane to the other side, okay, will have one particular another characteristic as per Gibbs Donnan membrane equilibrium, okay, law. That is that the product of diffusible ions on one side of the membrane will be equal to the product of diffusible ions on the other side. Okay, so it means that mathematically we can write it that the number of cations in solution A into number of anions in solution A must be equal to the number of cations in solution B into the number of anions in solution B. So product of their cation and anion in one side will be constant, in any side will be constant. That is a special thing found in our living organism. That is a according to it is described by Gibbs Donnan membrane equilibrium, okay, condition. So that once this thing happens, now what happens? Something new we will find. What is that? Consider that in one side of the solution, consider that in one side of the solution, there is one anion that is placed called anion X. X minus means anion X. It is a non-diffusible anion. It is inserted there somehow. It is inserted there somehow. Then the whole thing will be completely disturbed when some anion X is present in one solution. What will be there? The electrical neutrality will be maintained here also. No problem. So it will be what? The total number of potassium ion in solution A, that is the positive ion or cation, is equal to the total number of anion here. That the total number of chlorine ion and the anion X. So constant. So the total number of K plus is equal to the total number of anion Cl minus and X minus. So the number of potassium ion in solution A is higher than the number of chlorine ions. Number of potassium ion in solution A is higher than the number of chlorine ion. And in solution B, what happens? The total number of potassium ion must be equal to the total number of chlorine ion, that is the anion. They are equal because there is no such X ions present. Now, X is not a diffusible ion. The only diffusion is possible by potassium and chlorine, not by X anion. So the product of diffusible ion will be what? The potassium ion in the solution A into chlorine ion, anion into solution A. Their product will be constant, will be equal to the number of potassium ion in solution B into number of chlorine ion in solution B. But I told you that the potassium ion is larger in number than the chlorine ion. Okay. So in other way around, so potassium ion is larger than chlorine ion here. So therefore you can say that the potassium ion is larger than chlorine ion. So ultimately, if you see the previous thing, we'll find out that potassium ion in solution is more than the potassium ion in solution B, because the total number of ions are same to get electrical neutrality. So in solution A, there will be more potassium ion than the potassium ions in solution B. Similarly, the number of chlorine ion will be more in solution A than the chlorine ions in solution B. So that is happening. So this way it will happen. So that's how. So ultimately what happens, therefore, as the charges are distributed differently in two sides of the solution, some voltages will be there. Okay. So this is called, okay. So the diffusible anion concentration will be greater on one side, okay, without diffusible anions. So that is the thing that is mathematically present, is written here. This is called Gibbs Donnan equilibrium condition. Okay. Now once such different amount of ions are distributed now, okay, in the different cell part of the solution, solution A and B, some voltages will be manifested around the boundary of the two solutions. Some voltages. Now the question is, what will be the amount of voltage? What will be the amount of voltages? that we can find out. Okay, it is given the amount of voltage generated due to the differential amount of charge separation is given by an equation called Nernst equation. It is okay, Nernst equation. It is what if that gives us the Ex equal to that particular relation, RT by ZF ln logarithm to the base E. Okay, concentration of okay X in solution 1, ion concentration 1 divided by the ion concentration 2, okay, in the solution X. So the thing is that here R is the gas constant, T is the temperature in Kelvin, Z is the valency of the ion or valence, and Faraday constant is F. Ex is the amount of voltage or the potential that is manifested under equilibrium condition, okay, due to the ion X, okay, due to the ion X. Now X1 is the concentration of X in solution 1 divided by concentration of X in solution 2. So due to that ion X, how much is the voltage? That is actually found from that particular thing. Now see just one example I am telling you. At a normal body temperature, what is our normal body temperature? You know it is 98.4 or 98.6 degree Fahrenheit. Say if we consider 98.6 degree Fahrenheit, if you apply the formula C by 5 equal to F minus 32 by 9, then put F equal to 98.6, you will get C will come out as 37 degrees Celsius. So our normal body temperature is 37 degrees Celsius. So at normal body temperature, if you put okay the different values of R, T, and F, R is the gas constant, T temperature, all such things. Okay, so the formula is coming something like okay minus 61.5 log C in by C out. That is, C in is the concentration in the inner part of the cell, C out is the concentration in the outer part of the cell. So as an example, 61.5. Now just one example is given in a mammalian muscle cell. Okay, if the potassium concentration is 155 milliequivalents per liter, milli m eq per L means milliequivalents per liter in the intracellular fluid and 4 milliequivalent per liter in the extracellular fluid. We know that normally potassium ion exists in the inner part of the cell. It is not present in the outer part of the cell in the normal condition too much. So C in will be 155 and C out will be 4 concentration. C in will be 155 and C out will be 4. We put it, finally our equation comes as minus 97 millivolt. That is the voltage generated, okay, okay, within the cell due to the potassium ion. Okay, it is a voltage generated in the membrane due to the potassium ion. Okay. And similarly, if we calculate it for chlorine ion and sodium ion, we'll find out just by applying the same formula, we'll find out that say for sodium ion, it may be around plus 65 millivolt. It is mostly present in the outside of the cell. And chlorine ion, it is mostly present in the outside of the cell, but it is একটাই ছিল, দেখলাম আরেকটা কোথা থেকে লাগিয়েছে বুঝলাম না কি।
2. মাথা বড় হয়ে গেছে
কি বলে পড়ে গেল
কিছু বই উঠেছি বইটাকে বই উঠেছি না ঘুম করো কেন বসো বসো চুপ করে বসো যাও ঘুম করো চুপ করো ঘুম করো হ্যাঁ ঘুম যাও তো হ্যাঁ ঘুম যাও ঘুম যাও
So let's start our discussion again.
চিকেন 500 টাকা নিয়েছে আর জানতে
হ্যাঁ কিছু নেবে জানতাম কত জানি না 2000 দিয়েছিলাম Okay, let's start. So again, so that's how due to the chlorine ion or anion mostly present due to in the outside of the cell, it is minus 90 volt typically is present. Due to the sodium ion mostly present, there is a cation in the outside of the cell, the voltage is 65 millivolt manifested. And due to the potassium ion, it is minus present in the inside of the cell, it is minus 95 millivolt. So different ions are present in different amount, and due to that, okay, different voltages are there. So total voltage is actually the algebraic sum of all such voltages around the cell membrane, okay, at any point of time. So there are different examples are given, okay. So equilibrium potential for important ions in a neuron. Neuron means nerve cell. But there are the Nernst equation cannot predict all the voltages accurately because there are some demerits of this Nernst equation. It works only when the membrane is permeable to a single ion. But multiple ions are present in the system and they are having different permeability, different amount of permeability for different type of ions offered by the cell membrane. Okay. So sodium, potassium, chlorine, also other anions also there. Okay. And the conductance of sodium and potassium ions are also especially important to make the neuron work. So ultimately Nernst equation cannot work quite accurately. Okay, cannot work quite accurately. So to eliminate that problem, another equation was proposed that they called Goldman-Hodgkin-Katz voltage equation. Normally it is called Goldman equation. It is the modification. It is a remarkable modification in that particular field. And due to that new equation called Goldman equation, okay, people, okay, they got Nobel Prize. They got Nobel Prize for introducing an equation. But we are not introducing that equation in this particular class. I introduced it earlier in my class, but later I deleted. In the very beginning, I stated a little bit very big in the beginning. Okay, but right now I don't want to say this type of thing because we will deviate from our discussion. Okay, that's why I just mention that this equation can predict these voltages quite accurately, very accurately. That is all required for our purpose and nothing more. So the Goldman equation. So that is ultimately, so what is the essence of the discussion? The essence is that ultimately we know that across the cell membrane, we have some voltages, and under normal condition, the voltage is negative inside the cell. It is called resting potential. Okay. And that due to the intercellular biochemical processes, various chemical reactions running due to various ionic transfer, under normal condition also, the cell membrane is attaining a negative voltage. Okay. And it is found that normally sodium ions have the tendency to move from outside to the inner part of the cell whenever a favorable condition comes. Not always. Normally sodium ion mostly present outside the cell, not mostly. But whenever cell membrane can offer a path under certain condition, then only it can move from extracellular to intercellular medium. For potassium ion also just opposite. Normally it is mostly present inside the cell, but whenever cell membrane offers a path, it can transport from inner part to the outside part. Same is true for chlorine ion that moves from outer part to inner part. Normally, it is found that the crossing of ions to cell membrane is in the ratio of 3:2. But if three sodium ions are penetrating into the cell, inner part of the cell, potassium ions are coming out, okay, by two amount, okay, from inner part to the outer part. As a result, under excited condition, due to the ionic transport, our cell becomes positive. Cell voltage becomes positive. Normally, our cell voltage is negative, our resting membrane potential is negative. But under some excitation, when sodium penetrates the membrane, potassium penetrates the membrane, then our cell voltage will become more positive. Okay, it will be positive. Okay, and it will carry positive voltage. Okay. Now, through which path the ions can move? There are two things. One called ion channel, another is called ion pump. Now, ion channels are the path through which ion can move from one side to the other side as per their concentration gradient. According to their concentration gradient means ions that are having higher concentration in one side will move through the ion channels to the other part of the medium, okay, from higher concentration gradient to lower concentration gradient. And that path is called the ion channel. That path is called the ion channel. But ion pump, it is just opposite. The ion pump, it is just allowing some pumping effect. Means when a lot of ions are moving from one part to the another part, like sodium from outer to inner and potassium from inner to outer, the task of the ion pump is just to do the reverse process. It will move the ions against their concentration gradient. Okay, so it will restore their original condition. But but we know the pump can work only when some energy is supplied to the pump, because it is moving against their concentration gradient. So where from the energy is obtained? The energy is obtained from the ATP, okay, of the mitochondria of the cell, ATP, adenosine triphosphate, where some energy is stored in the form of ATP. So mitochondria is the powerhouse, it supplies the required energy sorry, so that the ion pump can work and the ionic condition will be restored finally. Clear? Next. So schematically we can say that the channels can be opened or closed. An open channel means ion can move, can pass. A closed channel means ion cannot pass. Okay, so that is this thing. Normally a cell remains at negative potential called resting potential or resting membrane potential. In our body it is normally minus 50 to minus 100 millivolt. Normally it lies within minus 70 millivolt to minus 90 millivolt. Normally it lies within minus 70 to minus 90 millivolt. Okay, within that thing. So it is that particular normal resting potential. Now, when the cell voltage becomes positive, okay, due to the ionic transfer, okay, then it is called a depolarized cell. A depolarization means a cell voltage becomes positive. Now, when the cell voltage regains negative value, again we call it repolarization. It is repolarized. And when the cell voltage is more negative than its normal value, normal resting potential, it is called hyperpolarized, more negative. It happens also under certain conditions. Okay, hyperpolarized. So main thing is the polarization and depolarization. Polarization means negative cell voltage. Depolarization means positive cell voltage. You have to remember that thing, because I shall use this term quite frequently in various contexts. Okay, polarization and depolarization. Polarization is negative voltage, depolarization is positive voltage. Now, when the biochemical process will start, so that there will be a change in voltage, okay, it is started or triggered by a certain condition called a stimulus is triggered by a stimulus. In plural form, we call it stimuli. Stimuli, when multiple stimulus is there, it is called stimuli. Now, stimulus is meaning one in physiology. Stimulus means it's a detectable change in the internal or external environment. Just like our external condition is that touch or pain, vision, smell, taste, sound, equilibrium, electricity. These are the external stimuli, due to which our biochemical process inside the body will change. It will be triggered in a different direction. An ionic transfer will start, and there will be a change in voltage. Okay, there will be a change in voltage. Normally, the ions cells that are remaining at polarized condition, resting condition, now will become depolarized. The cell will become positively, okay, charged, or its voltage will be more positive. And the internal stimulus is the blood pressure, heart rate, cardiac output. That may change. Due to that thing also, some excitation may be there. Okay. So if there is a change in blood pressure suddenly, we don't feel well. Due to that, we just either sit, we do something, we take medicine, we do something. So there will be some excitation propagating through our nerve cell. Okay, and there will be a change in biochemical process, and our cell will be depolarized, and some information will be transmitted. Okay, after depolarization. So that will happen. And homeostatic imbalances means it is actually the homeostatic balance is the ability of the internal environment to remain in equilibrium despite any internal external changes or external environmental influences take place. But homeostatic imbalance is just opposite, means okay, any internal or external condition changes in the body. When internal condition changes in the body or any other external condition changes, our body cannot come back to its balance condition. That is called homeostatic imbalance. Just like due to the aging, when our age increases, due to the genetic condition also sometimes, okay, our body cannot restore easily. So homeostatic imbalance will come. Other things, that there are deficiencies in nutrition, good food value, physical activity, lack of physical activity, mental health. So when we are in anxiety or very much stress or depression, obviously our body cannot function properly. So it depends on our mental health. So we may breathe heavily, we may feel many other problems, the digestion may not be proper, so many other constipation may come, many other condition will be there. Our blood pressure may change, many other thing may happen. So this is also changing due to the mental health. And the drug and alcohol abuse are the other reasons. Okay, and some toxic effect, toxins are also playing their role, for which homeostatic imbalance will be there. So whenever such imbalance takes place, so that will serve as a stimulus, and that information will be propagated to our body. Okay, and our body will try to do something to its best. So that is the stimulus. That is the homeostatic imbalances. Okay. Now, when some stimuli come or stimulus comes, maybe internal or external, whatever it may be, how the information will be transmitted to our body. If we see in a short way, simple way, we'll see that we'll have the idea how things will happen then. That our cellular response to stimuli will come into act. Okay, how it will be there? In terms of the movement, body movement, secretion of some hormones or enzymes, enzyme production, some gene expression will be there. Okay, and then some receptors. Okay, uh, our receptor on the cell surface, there are some chemicals called receptor on the cell surface that will sense the components that will the change actually that is coming due to the stimuli, and it would change, and ultimately the stimuli will be then what happens, the receptor will relay the information to the control center, means maybe to our spinal cord or to the brain, okay, for further processing and response. Okay, and some ionic transfer will start working through the cell membrane. There will be a change in voltage. So a chemical signal to voltage signal conversion will be there. Chemical to, due to the chemical effect, some voltage will be there. That is called a transducer effect, a transduction. This is called transduction. In our terms, it is a chemical to electrical conversion. The process of converting chemical signal to electrical signal via trans is called the transduction. So transduction, so what is the sequence of transduction? If we say this thing, here are the steps. Let's see. Our first thing is that there will be some change in pressure, vibration, light, when the stimulus, just stimulus will be there. Okay. Due to that presence of stimulus, okay, some chemotransmitter will be released from one cell. Chemotransmitter. For neuron, it is called neurotransmitter. For other cell, it is called chemotransmitter in general. But neurotransmitter, the term neurotransmitter is more common. Neurotransmitter is more common than chemotransmitter. So this neurotransmitter, okay, or chemotransmitter, it binds in some chemoreceptor. When the neurotransmitter is emitted in one cell, see here, when there are two cells shown in our figure, when a neurotransmitter is emitted from one cell, it will go to the other cell, adjacent cell, and it will be captured by another chemical present in the other cell surface. This chemical is called the receptor, chemoreceptor or neuroreceptor. Chemoreceptor or neuroreceptor. So a chemoreceptor or neuroreceptor will be combined with the chemotransmitter or neurotransmitter. So after such binding, okay, after some binding takes place between a receptor and transmitter, a new biochemical process will be started. A new biochemical process, a series of chemical reaction will start working. A series of chemical reaction will start working. And due to that chemical reaction, some ions will start transferring from one side of the cell membrane to the other side of the cell membrane. As ions are moving their place, okay, there will be a change in charge distribution surrounding the cell membrane, and due to the different charge distribution, some voltage will be manifested. So finally, as a result of some stimulus, some electrical potential will be manifested. This is called our transduction. Okay, so as a response to the presence of some stimulus, some voltage will be generated, and that process of generation of electrical voltage is called transduction. Right. Now, what are we know the different type of stimulus present. Different type of stimulus present. The next question is, where do we find this stimulus? What are the sites of having this stimuli? The site, the type of sites means the places where this stimuli can come. The stimulus will be present in the nerve cell or neuron. A stimulus may be present to the muscle cells. Then some muscle voltage will be found. A stimulus may be present in the endocrine system, means various gland system, so that different hormones and other chemicals may come out of the gland, and it will start triggering other processes within our body. The stimulus may be present to the digestive system inside the stomach or other part, for which different hormones and enzymes will be released, and our digestion will start working accordingly in a particular direction. So that is the different sides of the stimulus. Now next is the extracellular biochemical process. The thing is that in the biochemical process, actually, when our ion channels are moving, are actually allowing the ions to move, there are basically two type of ion channels, not two exactly, three type of ion channels, but for our purpose, only two ion channels are sufficient for our purpose. So one type of ion channel is called ligand gated ion channel, through which ions will transport from one side of the cell membrane to the other side, from extracellular to intracellular or intracellular to extracellular. So when the ion channels will open, the ion channels will open when some chemical signal called ligand, when some chemical signal called ligand, okay, starts binding to the to a part of the ion channel, then those channel will start opening. Some tunnel will be formed through which ion will move. The tunnel like thing will be formed and ions will start moving. So ligand gated ion channel will open in response to a chemical signal or in response to some ligand. That's why they are called a ligand activated or chemical signal activated channel, ion channel, or called ligand gated ion channel. Now, due to this ligand gated ion channel, when ions start transferring from one side to the other side, there will be a change in charge distribution. So, some voltage will take place, will be changed. And due to the change in voltage, now other channels will start working. Other channels. So, first the ligand gated ion channel will work, then other channel will start working in response to the change in voltage. So, these other channels that can respond to the voltage, new voltages, these are called voltage gated ion channel or a voltage activated ion channel. So these channels open in response to some voltage. That is when some cells are already getting depolarized. But for the first series of depolarization, we need the activation of ligand gated ion channel. Once that ion channel start opening, then charge transfer will take place, then some voltage will be manifested, and then these newly manifested voltage will trigger the successive voltage gated ion channels after this, and then the voltage gated ion channel will start working. So that is the method. Okay, there are two types of ion channel. Here is the shown that the ligand gated ion channel, some, okay, when some chemical, when some neurotransmitter is coming out of one cell and it is released and goes to the other part of the cell, other side of the cell, adjacent cell, some neuroreceptor will be there. So a neurotransmitter, when combined with the neuroreceptor, okay, will generate, after combination will be a chemical will be formed that is called a ligand, and in response to such combination, okay, or the ligand, okay, now the ion channels will start working. Ion channel will start working. The first ion channel that will work will be the ligand gated ion channel. So first neurotransmitter will be released from one cell, then it will go to the next cell, then some neuroreceptor will be combined with the neurotransmitter. After combination, we get a new chemical. This is called ligand, and that ligand will trigger ligand gated ion channel. Some ion channels will be opened. Ions will start transporting, and some voltage change will be there. And due to that voltage change, now the voltage gated ion channel will start opening. Okay, and that process will go on and on. Okay, in a constructive manner. So that is the method. That is the method of the whole thing. Now, so voltage gated ion channel, due to the some presence in voltage, okay, some the channel opens, okay, and some ions will start opening. So that is our voltage gated ion channel. So due to the change in voltage stimulus, some ion will start working. Now note that sometimes the thing is that whenever it may happen that in response to some stimulus, our ion channels may not start working under the effect of some medicine. Under the effect of some medicine, our ion channels may not start opening. So if the ions does not start opening, ions cannot start moving. So the voltage will not change. So no change in voltage means that external information will not be received or will not be detected by our body, or even when it is detected will not move to our brain or spinal cord. So it means then our body and mind will be quite free from external disturbances. So medicines, sedatives, various sedatives, sleeping pills are this type of medicines that are actually does not allow the ion channels to open under the presence of external stimulus. So as they don't open, there is no ionic transfer, there is no information transfer also. So our body and mind will be very much in peace, and we can sleep easily, and we can continue our sleep, sound sleep. So that is this particular thing. That is the essence of this type of medicines. Okay. But excessive taking of this type of medicine will harm, because it will, our body will depend on this type of medicines, and ultimately our body, what happens, cannot work in a normal way until and unless these medicines are taken. So our ion channels will be heavily dependent, its working will be heavily dependent on this type of medicines. Otherwise they will not be deactivated normal way. Okay, it will be deactivated only when the medicines are taken. It means what? Continuously will be disturbed by external stimuli, and we cannot sleep easily, we cannot relieve of our tension easily. Okay, so that's why excessive taking of medicine is not allowed for such reason. So that is the thing I am telling in simple way. Now there are, just I want to mention only one thing, I want to conclude with that thing, that so we know there are two states of ion channel. One is that either opening of ion or closing of ion, opening or closing. But there is one more state of ion, especially the sodium ion. For potassium ion, only two states, open and close. For potassium ion, in the potassium ion channel, it is just, if you see in the right side, there are open ion channel open and ion channel closed. That's all. But for sodium ion channel, there are three states. Between open and closed, there is one is more called the inactivation channel, inactivated ions. Inactivated means ions are as if open, but it is inactivated. Means the ions cannot move, cannot pass through itself. There is a subtle difference between opening and closing. Okay, here ions are cannot pass through this. Partly it may get trapped inside the ion channel, but it cannot complete its journey from one side of the channel to the other side of the channel. Effectively, it is a closing of the channel, effectively. So that is called inactivation state of the ion channel. It happens only to the sodium channel, does not happen to the potassium ion channel. So that is the basic things of that particular thing. So once we know this particular thing, now in the next chapter we can see how information will be transferred. Okay. Now let's see. I want to switch to. I don't want to say too much now in the very beginning. First class. Only thing is that let's see just our second chapter. I want to introduce now, just with such knowledge of ion channel, ion pump, okay, opening of ion channel, closing of ion channel, the concept of resting potential, which is non negative, the concept of polarization, depolarization, all such thing. We'll see how voltages are generated within the living cell. That we want to see. What are the steps? What are the steps of generating voltages within the living cells, and how these are there? We'll see also. One thing is that our biopotentials are having different types, two types. One is called as spontaneous potentials, the voltages that are always being generated inside our body continuously under the effect of some external stimuli, always. Another is the evoked potential, when some special external conditions are coming, then only our body will generate some special voltages. Okay, not the voltages under normal condition. These are called evoked potential. Let's see this thing. But first we want to go to the spontaneous potential. Spontaneous potential. Now, what is this spontaneous potential? It is always generated in the body, just like EEG, electroencephalogram. Some pattern of voltage always available in our brain, always. Where when we are whether we are uh, in sleep or whether we are in active condition, always will get some voltage. The voltage pattern may change, depending on our activity and mental condition, what will always be found? ECG, electrocardiogram. It is generated in the heart due to the heart activity. Always it is generated spontaneously and continuously. EMG, electromyogram, the voltage in the muscle, it is always generated continuously. Okay. And EOG or ERG, electrooculogram or electroretinogram, due to the eye activity, eye movement and all such things, continuously the voltage is generated in the eye. Electrooculogram or electroretinogram. These are the spontaneous potentials. Get my point? And okay, these are the spontaneous potentials. These are the biopotential sources. Another type is the evoked potential. How we get it? An electrical response of nerve cell, a group of nerve cell, to externally induced stimuli, especially to determine whether or not an area of the brain receives sensory information from a particular stimulus. It means what? Let's see, it will be clear if I say the next thing. Say, our evoked potential is classified into several ways. One is called sensory evoked potential, motor evoked potential, and event related potential. Our sensory evoked potential is that those voltages, those class of voltages that are generated inside our body or inside our brain, particularly in response to some external effect. When we see something, okay, and then some brain voltage changes, then it is called that new brain voltage is called EG. That particular voltage is called visual evoked potential, VEP. Visual evoked potential, VEP. When we hear something and our activity of brain changes, the EG is getting different in our body, in our brain, then it is called auditory evoked potential, AEP. Okay, in response to some hearing effect. Somatosensory evoked potential, the voltage which is generated inside our body or inside the brain, okay, in the form of EEG, as a response to some touch. Somatosensory means as a response to touch. Okay, evoked potential. Next is, let's see, VEP, visual evoked potential. See that what? Our brain is having different parts like frontal lobe, parietal lobe, occipital lobe, temporal lobe, different parts are there. Spinal cord is also going there and cerebellum is there, different parts of the brain. Different parts are handling different things, okay, of our brain. Now, when there is a visual stimulus, okay, just like say in a computer screen, something like an alternating checkerboard pattern is there. So a change happens in the computer screen. Then obviously some EG will be found, okay, in the brain. Okay, so some electrodes will be placed properly on the back of our head, and some voltage will be observed as a reading, okay, in the form of EG. So it is a visual evoked potential. In response to seeing something, that voltage is changed. Auditory evoked potential is again that in response to the presence of some sound as stimulus in the form of different speech, frequency, tone, all such things. Okay, okay, then some change in voltage will be found in our EEG. The responses are recorded from electrodes that are placed on the scalp of the head, the skin surface of the head, and are observed as a reading on an EEG, electroencephalogram. That is called auditory evoked potential. Our somatosensory evoked potential is a potential as a in reply to a touch. Actually, there are nerves running in the leg called tibial nerve, and sciatic nerve is also there. Sciatic nerve, okay. So different nerves are present. In our heart also, in our hand also, different nerves are there. Median nerve, ulnar nerve, different things there. So when some person touches us, or we are getting touched by some inanimate object also, we can feel the touch, and then obviously some response will be found in the patient's scalp, in the head, okay, or in the palm also sometimes it is found, okay. So it is recorded then, that voltage. So that voltage is actually the information of touch is carried by the tibial nerve, median nerve, ulnar nerve, all such things. So this touch information is called somatosensory evoked potential. The voltage coming as a result of some touch. Somatosensory evoked potential, ACP. Motor evoked potential is just opposite thing, that uh, here it is not coming as a result of touch or hearing or seeing something. Okay. It is coming as a result of the stimulation will be in the form of electrical or magnetic signal. Okay. If the electric stimulus is thrown to our body, it is in the form of a constant current high voltage pulse. Okay. High voltage pulse is 400 volt here, 400 volt AC say, or approximately 400 volt. A current will be 0 to 1 ampere, 0 to 1000 milliampere. So 1000 milliampere means quite a high current for our body, because we can get shocked just heavily in presence of only 1 milliampere current. In presence of only 1 milliampere current. So 1000 milliampere is quite a large current in that respect. And when some electrical stimulus is present only of a short duration, only up to 2 to 50 millisecond, 2 to 50 millisecond. Okay. So our electric stimulus is present only for short duration, 2 to 50 millisecond, in the form of a high voltage constant current pulse of 400 volt and 0 to 1000 milliampere. Okay. Then some voltage will be found. Okay. A magnetic stimulation is in the form of rapidly changing magnetic field. The field strength is really, flux density is very high. It is 1 to 3 Tesla. Tesla means Weber per meter square. You know, 1 to 3 Tesla means quite a high magnetic field. Okay. And frequency is what? The alternating magnetic field in the range of, which frequencies in the range of 5 to 50 hertz, and only for a short duration, 50 to 200 millisecond only. It's a magnetic stimulation. So our electric stimulation or magnetic stimulation will be applied centrally on the spinal cord or on the scalp of the brain, and it will be recorded peripherally in distant muscle. So it will be captured at some remote location, okay, at some distant muscle. Okay. So the impact of the muscle behavior will be studied, and it will be decided whether it will be judged whether the information can be transmitted easily throughout the nerves and the pathways. Okay. So motor evoked potential. And our last thing is the event related potential. I want to conclude today with that particular thing. Actually, the cortical activity is recorded, the activity of our brain actually recorded. Okay, but here mainly it is related to the stimuluses with respect to our cognitive stimuli. Means by understanding something. Okay, in relation to some cognitive excitation, it will be there, and the stimulus will be coming quite infrequent manner, not coming regularly. Okay, so that is our event related potential ERP. When we understand some things, we are having some emotion. Okay, so then this type of voltages are coming. ERP. Just the comparison is that our event related potential is of the order of 1 microvolt to few microvolt, not too much. EEG is having higher tens of microvolt electroencephalogram. Just this is our last slide today, but we are forced to start it again. So just for comparison, our EMG is of the order of millivolt and ECG it is of tens of millivolts, somewhat larger. So that is a brief comparison of various magnitude of voltages present in our body. Next day we'll start discussing how our voltage is getting generated in one part of our body and is getting transmitted to the other part of the body through nerve cells or neurons as a result of the presence of some stimulus. Okay, and some more aspects will be studied in our next class. আচ্ছা, বিফোর দিস ক্যান ইউ টেল মি ওয়ান থিং? আজকে এটা হয়ে গেল, আমি তোমাদের মেটেরিয়ালটা পাঠিয়ে দেবো, কোনো